her master's in genetic counseling in 2004, Kelly Hagman spent several years in a research lab studying DNA mutagenesis. She started her career as a genetic counselor at the Molecular Diagnostic Laboratory at the City of Hope, where she continued her involvement in research, most of which focused on Li-Fraumeni syndrome. Driven by her interest in new molecular technologies, in 2009, Kelly joined Ambry Genetics to lead the Genomic Services Group, where she gained expertise in multiple sequencing platforms, DNA and RNA enrichment techniques. In 2011, she transitioned to the clinical team to lead Ambry's clinical genomics offerings, most notably diagnostic exome sequencing. Kelly is currently the co-chair of the National Society of Genetic Counselors Genomic Technologies Special Interest Group. Kelly is also a firm believer that the best patient care is achieved through open data sharing. She has more than 35 peer-reviewed publications in molecular genetics and has been an invited lecturer at several scientific meetings. We're very lucky to have Kelly speaking with us today. Our next speaker is Erica Smith. Dr. Erica Smith received her BS at the University of California, San Diego in animal physiology and neuroscience, and PhD at the University of California, Irvine in neurobiology and behavior in an Alzheimer's lab led by Dr. Carl Kottman. Her thesis established the molecular response of hippocampal neurons to simultaneous growth signals, such as those from brain-derived neurotrophic factor, BDNF, and inflammatory cytokines. Dr. Smith then completed postdoctoral research in a blood-brain barrier lab with Dr. Drayton Aguli, studying an animal model of pandas. She also studied the effects of repeated strep throat infections on mouse brains. At Ambry, Dr. Smith specializes in evaluating clinical validity, or the evidence linking genes to specific diseases. We're very lucky to have both Dr. Smith and Kelly here today with us. And with that, I'll go ahead and give the mouse to you.
Thank you, Leila. Okay, so I'm going to start the presentation by doing part one, which will be a history and evolution of clinical exome sequencing at Ambry since its launch in 2011. And through that, I'll be going through a timeline of events that occurred and lessons that we learned. And through that, I'll talk about our variant filtering pipeline, our variant classification system that we use ubiquitously at Ambry, as well as our reporting criteria as it relates to exome sequencing. I'll also go through our data sharing efforts and the lessons that we've learned from analyzing and sharing that data that has led back to improving our product. Following that, Dr. Smith will go through parts 2 and 3, which will be highlighting 2 of our most recent publications that we're really most proud of. They're probably our most landmark publications that talk about our processes. One is our clinical validity scoring system, which describes how we classify gene-disease relationships. And the other one is our criteria for candidate genes and how both of those actually relate to our patient for life philosophy, which leads to reclassifications for patients. Okay. So you'll see the timeline here. So 2011 is when we first launched Exome. We're very proud to be the first lab to offer diagnostic exome sequencing. We, from the very beginning, have set our sequencing and informatics filtering standards to be very high. So we achieved greater than 95% of the exome covered with at least 20x. And our informatics filtering pipeline from the very beginning was set up to be phenotype and inheritance pattern agnostic. And I'll talk a little bit more about what that means and why that's so important. It also, our pipeline has also been tailored to reduce false negatives. So what that means is we put the onus a little bit more on our exome analysts to do a manual review of alterations. We want to make sure that we're not filtering out anything that could be relevant. Okay, so I'm going to take us through just kind of a crude overview of our bioinformatics filtering pipeline without getting too granular. This will show us how you get from roughly between 200 and 400 annotated variants per patient down to the potential 1 or 2 that could be relevant for that particular patient's phenotype. First, the biggest steps are that we will actually filter out alterations outside of the coding region, and we filter out non-splice related synonymous alterations. That will get rid of a lot of alterations. However, we will always actually protect alterations that that we have deemed in our database as founder mutations. Those will not get filtered out. Any alterations that have an HGMD mutation ID, as well as alterations that we've classified internally at Ambry as mutations or variants likely pathogenic. Then we go through our family history inheritance-based filtering. It uses inheritance models, so it'll filter variants as if, for example, in a patient where there's an affected proband and mom and dad are unaffected, in the autosomal dominant model, we would look at only de novo alterations. In the recessive model, we would look for alterations where one alteration was inherited from each parent or one was de novo. Along with that, we always run what we call a proband-only model, which is basically a reduced penetrance model. So this will look at alterations that were identified in the patient regardless of whether they were inherited from the parents. And you'll see that through this entire process, there's no filtering that's based at all on phenotype or likely phenotype that the patient has. That part is all done by manual review. So this will get us down to between 50 to 300 variants that the exome analyst will then review in a manual way. All of these alterations will get binned into 4 different categories. The first will be noise, so any artifacts or polymorphisms that survived filtering will get filtered out by the analyst. They'll also look for clinically irrelevant alterations. So these are alterations unrelated to the referral indication. Generally, these would be considered secondary findings. They would be reported if the patient had opted for secondary findings and they were mutations or VLPs. Then we have, of course, the relevant findings, and these are alterations that are likely or possibly the cause of the patient's referral indication. And then we have notable findings, and these are alterations that are unlikely related to the patient's phenotype, but there is some level of clinical overlap. Most of the time, these are alterations that can be likely benign, or there's really just some very non-specific overlap with the patient's phenotype. But we do feel it's important to include those on the report, so both of these categories do show up on the report. And in a minority of cases, we do get some, the clinicians are interested in the notable findings, and they'll come back to us and say, hey, we looked further and the patient has some additional features that are related to this finding that you had reported. Let's look at this a little further, and we can, and that sometimes we'll go back and look at it and can result in an amended report. Okay, so after our first year of sequencing exomes, our first 3 results came in towards the beginning of 2012. 2 of those, they were all 3 positive, so we had an awesome detection rate of 100% at that point in time. 2 of them were novel candidate genes, and 1 was a newly characterized gene. So this actually set the stage for, sets the stage for what we're talking about today, and what Dr. Smith will go into in detail, that we realized, right away that we were going to need to start developing a standard scheme for gene classifications, in other words, clinical validity, and that we needed to develop a scheme for when to report novel candidate genes on a patient report. We also immediately set up an infrastructure to data share, and these are actually our 2 first novel findings that were later published, but we realized how important it was to data share. 1, because we're benefiting from the data that's out there. So there'll be a publication that will come out. We've had examples where a new publication comes out about a gene-disease relationship, and then the next day we're analyzing a patient that has a mutation in that particular gene. So we realize how important it is and what responsibility we have as well to share that data back with clinicians and other laboratories that are analyzing exomes, because it's the best thing for patients. So our early observations, as more results started rolling in, we realized that actually about 1/3 of all of our positive results are in these recently characterized genes. So genes for which we just understood the gene-disease relationship within the last 1 to 2 years. And so that's really very huge, and that actually is held through today. That still holds true. So we realized early on we need a team to review the literature on a daily basis. So we developed, and Erica, Dr. Smith heads up our clinical validity team. to review the existing literature regarding gene-disease relationships, evaluate whether there's enough evidence to call gene-disease relationships characterized, and the ultimate goal is to take all the genes in the genome and categorize them as either characterized or uncharacterized. Also, some of our early observations that of all of the patients that send in samples, around 7 to 8% of them actually have an alteration in a novel candidate gene. So we realized also early on that we need an assessment criteria. So we got to working right away on putting together this assessment criteria. This was actually not published. We finally finished putting it all together and perfecting it, and was published in 2016. Dr. Smith will go into it in a little bit more detail, but in general, it uses animal models, in vivo and in vitro, protein co-localization information, expression data, gene disruption experiments, and etc. And then just to highlight some of our data sharing efforts. So right away, the easiest way to get data out there was to submit abstracts, talks, and posters at meetings. So we started doing that right away, and we've done that at every meeting since. We share all of our data to ClinVar, as well as all of our novel candidate genes and genes that are in newly characterized genes to Gene Matcher. And we actually have been involved in multiple collaborations with clinicians and other laboratories to work to further delineate phenotypes for particular genes based on Gene Matcher connections. And we also, apologize for the delay. Okay, so we also have multiple novel candidate gene research projects. So every time we report out a novel candidate gene in a patient, it's an uncertain finding overall, but we tell them that there is a multitude of evidence that we describe in the report that says that this is a potential new gene-disease relationship. Further research is required. And we go through every effort to connect those clinicians with researchers. We have multiple relationships with wet labs across the world. Highlighting a couple of these, we have ongoing collaborations with some institutions, including Jackson Labs for mouse models, and they've actually developed a couple of mouse models based on our candidate genes. And also we work closely with UCI for zebrafish studies. And then these are just a couple of examples, and there's multiple others. And then we also have our peer-reviewed publications. So we feel that in addition to data sharing in databases and abstracts and talks at meetings, it's very important to get the peer-review data out there. There's something about looking at your data and analyzing it that really pulls different things out of it that has later translated into improving our processes and improving the way we report and the way that we analyze data. So from the Exome group, we have over 40 peer-reviewed publications that is since 2011, since the launch of the product. We also are always open to any and all collaborative publications with clients, and we have multiple collaboration agreements with dozens of institutions and organizations all across the world. And then 2013, we were the first to publish our secondary findings experience by Shamrazadi et al., who's sitting here with us. So her paper highlights that patient decisions for disclosure of secondary findings among our first 200 patients that had ordered exome sequencing. And we found that most patients opt in. Most patients do want to get a copy of their secondary findings report and information about potential alterations that could affect them that are unrelated to the reason for referral. Multiple other articles have corroborated this since. We also realized that their patients really wanted options for expanded secondary findings, so we worked hard to offer, in addition to the ACMG standard 59 genes, we also offer multiple expanded options for secondary findings to patients. Also in 2013, we were the first to observe our expanding phenotypes phenomenon, and Leila Shamrazadi also presented this for us. It was accepted as a platform presentation at ACMG in 2013 called Expanding Clinical Phenotypes, Exome Sequencing Reveals Much Wider Phenotypic Spectrum Than Do Most Clinical Diagnostic Criteria. So this was, these observations confirmed our initial philosophy about our filtering pipeline to make no assumptions. So we do no filtering based on phenotype. So because of phenotypic expansions, these are positive findings that are in genes not traditionally thought to be associated with particular features. So if we had added any phenotypic filtering in our pipeline, we very likely would be filtering out some of these phenotypic expansion examples. Okay, so 2014 is when we published our original cohort paper, and this was to talk about our first 500 patients and the observations that we found. First was that overall everyone wanted to know what's the detection rate for exome. It's roughly 30%. That's roughly held true through today. It's gone down probably very slightly just because we've have already worked through some of the low-hanging fruit patients that have been waiting many, many years for a diagnosis, but it's roughly right around there. And we also observed that this rate has more than doubled the rate achieved with traditional methods. So exome sequencing is obviously an indicated and valuable test in many cases. Also, there's a higher detection rate with trios, and this data has been corroborated multiple times as well. There's actually a statistically significant difference, and it's quite dramatic, the difference in the rate of detection in trios versus proband-only cases. And then 2015, we published our experience among diagnostic exome sequencing in patients with a history of consanguinity. We found that actually over 38% of positive results are not an autosomal recessive pattern. There is a little bit of ascertainment bias there in that patients would have already gone through multiple autosomal recessive testing that was indicated, but it's still a very surprising number, also confirming our philosophy in our pipeline to not make assumptions, not assuming anything based on inheritance patterns. So we always would run this proband-only model and run every single type of inheritance pattern for every single patient. And most of these actually were de novo alterations that were found in these patients. And then in 2016, so now we're getting to what we're going to focus on, what Dr. Smith will focus on today is our two, what we consider our seminal publications. First is we were the first to publish our standardized algorithm for reporting candidate genes. And we found that 8% of patients that were sent in for diagnostic exome sequencing had an alteration in one of these genes. So we then have now finally published a scheme we've been using for many years, and it's an evidence-based criteria scheme for reporting novel candidate genes. And then 2017, we were the first to publish our classification of disease genes, or in other words, our clinical validity assessment. This is Dr. Smith's paper that came out in 2017, classification of genes, methods, standardized clinical validity assessment of gene disease associations, aids diagnostic exome analysis and reclassification. We now have a published standardized method for gene disease classifications and a devoted team that's a team that's devoted to these clinical validity assessments on a daily basis. So I want to briefly go through an overview. This is a roadmap to our reporting structure at Ambry and how important that clinical validity assessment and our novel candidate gene disease criteria plays in this roadmap. So first of all, on the left we have our clinical validity assessment. These are gene disease correlations and classifications. This is all based on the literature, and it's based on evaluation of all previously reported patients. So now when you look over at exome sequencing reporting criteria, that's all patient specific. So those that are on our clinical validity team actually spend their days analyzing the literature separate from patient samples that are sent in. All of the alterations, all of the gene disease relationships get categorized into to either no reported patients all the way up to definitive gene disease relationships. We have a cutoff between limited and moderate. Those that are moderate or above, we consider a characterized gene disease relationship. And then for these, anything that's limited or below, we consider an uncharacterized gene disease relationship. So for characterized gene disease relationships, our reports will describe the level of clinical correlation between what the patient's symptoms are and what we know about that gene disease relationship. And it will also describe the alteration classification, and then the overall results category will be a combination of those two. If either one of the two is uncertain, the overall results category will be uncertain. For our uncharacterized gene-disease relationship, we use our candidate gene criteria scheme, and we can categorize them into 3 different categories, either insufficient evidence to report out for a patient, or there's 2 levels that get reported, candidate or suspected candidate, and candidate has the highest level of evidence. And these will all be reported out as candidate genes of uncertain significance. And then I want to briefly take us through our variant criteria at Ambry. This is something that we use universally at Ambry, but when we analyze every variant, whether it's a panel test, exome, single gene test. So this is how we analyze variants. So we have, we use our Ambry database data, so co-segregation information, mutation co-occurrence, phenotype data, and then we also use published data such as case reports, functional assays, conservation data, etc., as well as population databases to give us a gauge of allele frequencies. These all come into play in our assessment scheme. This is available on the website to view. We have our criteria listed here. There's a point system, and you can get enough points to add up to a class from 1 up to 5, 1 being benign and 1 being a pathogenic mutation. We've analyzed the confidence for these alterations. Anything that we call a mutation has a greater than 99% confidence interval, and anything that we call a variant likely pathogenic has a greater than 95% confidence interval. All of these criteria were based heavily on ACMG and AMP guidelines and improved upon by our own criteria. And for variants of uncertain significance, we also have a family studies program. We have our functional studies lab known as our ATG lab, or Ambry Translational Genomics Lab, where we do multiple functional follow-up studies for patients that had a VUS, and then also we're starting to do this as well for patients that had negative exome sequencing or some other follow-ups or other cases where there was a finding where follow-up studies would be indicated. If we find anything and then we can confirm it on the DNA level, we would send an amended report back to the clinician. Our entire variant assessment classification scheme was described in detail in this paper from 2016 in the International Journal of Breast Cancer from Pesseron et al. We recommend that people will read that paper to get more in-depth information about our variant scheme. Okay, so across the top I have our whole timeline here, and I just want to end by my section just by talking about some of the cost savings papers that have come out. This is just a sampling of them, some of the probably the most prominent ones, and these are not published by Ambry, but they do all talk about the bottom line from all these papers is that 1, exome sequencing identifies 6 significantly more conclusive diagnoses than the standard care pathway. So one of them shows roughly 30% versus a 7% detection rate. Also, traditional genetic testing trajectory is 3 to 4 times as high as the cost of exome sequencing. So those are some of the bottom lines that have come out in the 5-6 years since exome sequencing was released. So I'm going to turn it over now to Dr. Smith to again go through in more detail our clinical validity scoring system as well as our novel candidate gene criteria.
Great, thank you. So I'm Erica Smith, and I'd like to describe some of the innovations that we use at Ambry to streamline and strengthen our clinical diagnostics reporting for exome sequencing. In this section, we'll talk about some of the recently published reporting criteria. Oh, it's a little delayed. Okay. But first, an overview of disease gene discovery. So we currently think that there are about 20,000 genes in the human genome, and we know about 4,000 genetic diseases. So in an egalitarian world of disease research, it would simply be a matter of figuring out which average of 4 or 5 genes cause each disease. But some genes, as it turns out, don't cause any disease. And some genes cause multiple diseases. So knowing this, we can predict that each gene-disease association will need to be carefully evaluated to make sure that we're accurately associating the correct pairs. Part of this is figuring out which genes are very tolerant to variation and which are less tolerant to variation. if we were to align all 20,000 human genes on a scale from the how essential they are for life, with the most essential genes on the left and the genes most tolerant to variation on the right, we would see that the function of the most essential genes, represented by this dark blue box, is so critical that we would not expect to see any viable humans with mutations in these genes, as predicted from animal models and recurrent human miscarriages. The genes on the right are more tolerant to variation, and we can easily find apparently healthy individuals with mutations in these genes. These genes just don't cause disease. In the middle of these two extremes, Chong et al. In 2010 estimated that there are between 10,000 to 15,000 disease genes. Of the disease genes, only about 4,000 are currently known to underlie disease, at least one Mendelian genetic condition. These genes are considered characterized. For characterized genes, we might expect to see multiple patients reported with mutations in the same gene and the same disease. And we usually have an idea of the mutational mechanism that causes disease. Often, diagnosing a characterized genetic etiology allows us to extrapolate from the disease course of previous patients to predict the disease course of new patients. Findings in uncharacterized genes, however, have insufficient evidence to confidently associate them with any specific disease. There may be few or no patients reported and limited functional studies. And candidate findings in uncharacterized genes are a unique type of result that is unique to exome sequencing. And because of the fast rate of gene discovery, genes are rapidly switching from uncharacterized to characterized. Depending on which paper you reference, between a quarter and a third of all positive findings from exome diagnostic sequencing are in newly characterized genes or discovered only 2 years prior to diagnosis. We can actually see the fast rate of gene discovery on this slide from this figure from the new Boycott et al. Paper. They graphed the number of new gene disease discoveries on the y-axis compared to the year on the x-axis. And you can see the rapid rate of gene discovery, about one every other day. Evidence that associates a specific gene with a disease can come in many forms, including human patients, functional studies, or animal models. With such a rapid pace of discovery, there's a clear need for a standardized evaluation criteria to determine which evidence is sufficient to provide a diagnosis. So, time for a quick quiz. What do all of these objects have in common? We've got a graduated beaker, a stopwatch, a scale, a thermometer, and the Wong-Baker Phases scale. I'm not going to wait for responses, but they're all tools for measurement. The beaker measures volume, the stopwatch measures time, the scale measures weight, the thermometer measures temperature, and the Wong-Baker Phases scale measures pain. We need ways to effectively communicate the amount of something in multiple domains of life. Along a similar vein, we need ways to effectively express how strongly a gene is associated with disease, and a standardized measurement tool for expressing that weight of evidence. In 2014, Biesecker and Green suggested using the term clinical validity to refer to the determination that a particular disease is truly caused by pathogenic variants in a particular gene. So I'd like to discuss a scoring system to measure clinical validity. This was largely inspired by some guidelines published in the MacArthur et al. 2014 paper suggesting what categories of evidence are useful to consider when evaluating gene-disease relationships. But even with these guidelines, it was still possible for different people to read the same papers and come to different conclusions about the amount of evidence underlying a gene-disease association. We all agree that reports of previous patients with mutations in the same gene and the same disease is the strongest evidence for a gene-causing disease. But what if only 1 or 2 patients are reported? In those cases of rare diseases, we may need to rely on evidence from in vitro or in vivo models. and it's helpful to know how much weight each of those categories of evidence should carry. So combining our communications with the curators of the clinical genome resource, the evidence from the categories from the 2014 MacArthur paper, and our experience analyzing over 4,400 cases since 2011, plus family members, we developed simple standardized scoring guidelines to determine the clinical validity of a single gene disease association. And this scoring system is in the paper that Kelly mentioned in the Journal of Human Mutation. It's the first peer-reviewed published manuscript for evaluating the disease association for individual genes. More recently, the ClinGen, the clinical genome resource, has published a clinical validity score sheet that's more exhaustive, but I'll discuss our scoring system today, and suffice it to say they both come to very similar conclusions. So in our score sheet, each category of evidence is allowed a specific number of points allowable, reflecting the usefulness of determining the usefulness of that piece of evidence for determining a gene-disease relationship. We optimize the points values available within each category over repeated rounds of testing and validation while scoring hundreds of gene-disease relationships. And before I get into details, I want to mention that this scoring system is intended for use in highly penetrant, early onset Mendelian diseases. It will likely need to be adapted for use in common or adult onset diseases like autism or cancer, but we hope it promotes discussion of standardizing evaluation of gene-disease relationships. So first off, and most important, we consider the number of previously reported patients. 1 point for the first 2 patients, a second point for the next 2 patients, 3 points for up to 9 patients, and 4 points for up to 24 patients. The points values reflect the diminishing return of simply identifying additional patients. For instance, there's often ascertainment bias in subsequent reports where researchers will specifically perform targeted sequencing in a disease cohort and identify alterations that may not actually be causing the patient's disease. Additionally, other forms of evidence, for instance, the mutational mechanism, are extremely valuable for diagnosing additional patients in the future. 1 point can be added for additional statistical evidence. For dominant disease, an excess of de novo mutations compared to expected for those genes can earn an extra point. For recessive diseases, extensive co-segregation of the variant up to a log score or above a log score of 3 can earn the extra point. In this example, you can see that we would normally count this as one family or one patient even for Mendelian genetics, but you see extensive co-segregation of this alteration in TECR with intellectual disability. In fact, there are enough informative meioses here that we can add this additional extra point for statistical evidence. We allow 1 additional point per publication for new publications that report additional patients to reflect acceptance in the peer review process. Also, additional publications are important milestones of replication, which is the basis of scientific discovery. We assign points for the number of variants that are predicted to be pathogenic or likely pathogenic according to published guidelines. This criterion is independent of the number of patients or the mutational mechanism or the inheritance model, but reflects the fact that variants that are predicted to be pathogenic or likely pathogenic are more likely to have a measurable effect on gene function than variants of unknown significance. There are 2 points available for evidence that the gene function is consistent with disease. 1 point for spatiotemporal expression is consistent with disease. So for instance, early expression of a gene in the brain would support a role in intellectual disability. A second point is available if the candidate gene product interacts with a disease gene product that's known to cause that disease. There are 2 points available for gene disruption, or evidence that disruption of the whole gene is relevant to the pathology of human disease. So the first point is for in vitro evidence that the whole gene disruption has a similar pathology. In this example here, you can see that the neuron expressing the wild-type gene has extensive dendritic morphology and a long axon. In a knockout neuron, you see truncated dendritic branching and a shorter axon. This might support a role in intellectual disability. The second point is available for in vitro experiments that reveal a mutational mechanism. In this case, we might try transfect or exogenously expressing the wild-type gene in a mutant cell that has a phenotype to test if it rescues the phenotype. For our second point. Finally, 2 points are available for informative animal models, which can be mice, Drosophila, zebrafish, or even C. elegans if they have an informative phenotype. The first point reflects evidence that the gene functions in vivo in a pathway relevant to the disease. So for instance, animals completely locking the gene the gene of interest might have impaired performance in a memory or learning task. The second point is available if the genotype and phenotype completely match the human disease. So this point would be relevant in our animal model if the animal model has the same gene dose as the human patient's, i.e. an animal model that is a complete knockout would only get the second point for an AR disease or recessive disease. The points for each category are summed and compared to guidelines for the clinical validity levels. You'll note that the definitive category appears to be off the chart. That's because we generally reserve this for genes that have at least 25 reported patients in them and for which we know the mutational mechanism. Below that, the categories of strong, moderate, limited, and no evidence have slightly overlapping categories to allow for clinical judgment. Genes with at least one gene-disease relationship of moderate or above are characterized, as Kelly mentioned earlier. And genes with less evidence for a role in disease should be considered uncharacterized or candidates. With the rapid rate of new gene-disease discovery, we should expect to continually reevaluate gene-disease relationships. So let's go through an example to see what it looks like. The gene THPO encodes the protein thrombopoietin, which promotes blood cell proliferation. The disease thrombocythemia has been associated with dominant mutations in THPO that are gain of function. And symptoms of it are excessive bleeding or hemorrhaging. There have been at least 3 splicing mutations reported in 5 families with thrombocythemia in at least 5 papers. And in 1999, Ghilardi et al. Reported that cell lines transfected with the mutant THPO cDNA overexpressed THPO protein compared to cells transfected with wild type. So if we sum up the points, we can get 3 points for the number of unrelated patients, which is 5, 3 points for the number of publications, 3 points for the number of variants predicted pathogenic, 1 point for gene function, and 2 points for gene disruption. This sums up to 12 points, which fits well into the category of moderate, making the gene THPO characterized. But then we had a patient with biallelic alterations in THPO and the opposite disease, aplastic anemia or thrombocytopenia, meaning not enough blood cells. Reviewing the literature, we found a report of recessive inheritance of a loss of function alteration. Dasuki et al. In 2013 reported 2 siblings with anemia, and when they transfected cells with the mutant cDNA, the mutant cells produced less THPO protein than the wild-type cells. Additionally, a knockout mouse has similar symptoms. So when we scored the evidence here, we got 1 point for number of patients, 1 point for number of publications, and then a point each for function and disruption, and 2 points for a matching model organism. This sums up to a total of 6 points, which results in a category of limited, meaning that this patient received a candidate report in a characterized gene. So this example demonstrates that each gene can be associated with multiple different diseases, and each disease can have a different clinical validity score reflecting the underlying evidence. At Ambry, we use this scoring system to analyze the clinical validity of each gene-disease association. In the process of reviewing new literature, we often move genes to characterized, And whenever we do that, we review patients with rare alterations in that gene to see if there's phenotype match. So to conclude this section, I want to point out that our published clinical validity system is a tool for evidence-based quality genetic testing. We use it in diagnostic exome sequencing to determine the type of report, either characterized or candidate, and then we also can use it to guide diagnosis of previously unsolved cases, because whenever a gene is switched to characterized, all patients with rare alterations in that gene are reviewed. This often leads to Ambry-initiated reclassification reports, which I'll go into in a minute. The clinical validity scoring system can also be useful for creation of disease-specific panels. We can create smarter multi-gene panels by prioritizing genes known to cause disease. In this last section, I'd like to discuss the types of case reanalysis and reclassifications within Ambry. Many cases that are reclassified start out receiving uncharacterized candidate gene reports, but you might ask, why look for findings in novel candidate genes? Well, it's actually been reported that candidate results in uncertain genes can lead to changes in disease management or intervention. New clinical management strategies have been reported for patients with candidate findings. And as we continue to uncover additional rare diseases, reporting candidate findings combats the N of 1 problem, where extremely rare diseases may not be observed again for a very long time. In that context, waiting to report a patient until additional patients are identified may either take several years or may never occur. Additionally, reporting candidate findings allows patients and clinicians to participate in disease discovery. Many of them have been under a diagnostic odyssey for years and are very capable of contributing to disease discovery, especially through connecting on a gene-specific basis through organizations such as GeneMatcher. So, as Kelly mentioned, in addition to our published clinical validity evidence, we also follow our published candidate gene guidelines for determining which genes can be reported out and at which level of confidence. The novel candidate criteria evaluate the same forms of evidence as the clinical validity score sheet, including previously reported patients, animal models, in vitro evidence, and evidence about the gene family. And again, these are published in hopes of starting conversations about standardizing reporting across different diagnostic laboratories. This paper reported that in our first 1,500 patients, candidate findings were identified in about 8% of patients that were examined. And interestingly, novel candidate findings are not only found in uncharacterized genes. Nearly 8% of our novel candidate findings were new phenotypes for a characterized gene. So a phenotype expansion sometimes, or even a new disease. So specifically, we say that we found 8% of our examined patients had candidate findings because novel candidate analysis can really only occur in a subset of patients. These are patients for whom a full diagnosis was not found in the characterized genes, and patients that have an informative trio. But because of the stringency in reporting criteria, we found that more than half of our novel candidate reports are subsequently corroborated in the literature. And whenever enough corroborative evidence accumulates to switch a gene to characterized, we issue reclassification reports for all previous patients for whom new information provides a diagnosis in a characterized gene. So this brings us to the last topic, reclassifications. There are two paths to a result reclassification at Ambry Genetics. The first is a clinician-requested reanalysis, which we've offered since February 2013. The second is an Ambry-initiated reclassification, and as far as I know, are the only lab that proactively reclassifies reported patients. So for a reanalysis request, we will reanalyze our cases at no cost with no time limit. If we look back at the cases from 2016 that received a reanalysis request, about 12% of those cases resulted in a change in result. we can see that there are a variety of reasons contributing to a change in overall results, including new clinical information provided by the clinician, new disease gene-related information for characterized or uncharacterized genes, new variant information, or even pipeline upgrades. I want to point out in particular that we recommend requesting a reanalysis whenever there's new clinical information, such as the development of new symptoms, or when following a variant of unknown significance. Now, while only 12% of reanalysis requests resulted in a change in overall conclusion, the other source of reclassification reports is only issued due to a change in overall conclusion. And in 2016, we issued 46 of these. And the vast majority were new diagnoses of previously unsolved cases. In nearly 80% of these cases, this was due to a newly discovered disease gene. And these patients are finally receiving their diagnosis, in large part due to database curation with clinical validity scoring. Here are some of the newly characterized genes for 2016 that led to Ambry-initiated reclassification reports. It's particularly encouraging to see that many of these genes were initially identified as candidate genes, the ones marked with an asterisk. And of all of the reclassification reports signed in 2016, you can see that the majority are older cases. This shows that we are continually evaluating and reviewing previous cases with no expiration date. And it also shows that over time, new discoveries are helping us solve more and more cases. Finding a diagnosis for our previous patients is good for them because it finally can give them an answer and an end to their diagnostic odyssey. We also think it's good for business because overall, looking at our 30% diagnostic rate, almost 10% are due to reclassifications. So to conclude, I hope that I've pointed out that retrospective analysis of previous cases significantly contributes to the diagnostic rate. We think that issuing reclassification reports is in line with our patients being patients for life, and we use published standardized reporting criteria to support diagnostic quality. Our experience emphasizes the importance of up-to-date literature review using a critical eye of clinical validity evaluation. And we still believe that data sharing is essential for progress. We're proud that we've submitted more than 10,000 variants to ClinVar, and we're continually publishing our findings. I'd like to thank you for listening and open up the floor to questions.
Great. Thank you, Dr. Smith and Kelly. That was a wonderful presentation and really talking about the importance of clinical validity to help us identify not only new causes of disease, but also, for the greater good of science. That was wonderful. We have a few questions here, and for others, feel free to type in any other questions you have. We have a couple questions. I'll kind of combine them about if a patient has had whole exome sequencing done or smaller panel testing done previously at another laboratory and whole exome sequencing is offered at Ambry, do you skip over those results, or like let's say they got a VUS or they had a limited panel of genes, do you skip over those or do you reevaluate all of those as if it were a new patient?
Yeah, so that's a good question. So we'll go through all of our analysis, as if, the way that we would normally for any other patient, and we would categorize all of the alterations into those 4 bins that I described. And then if there was if we were provided with a report and there was an alteration or a variant of uncertain significance or any other alteration that was identified previously, we'll include that alteration in a note section on the report saying we did identify this alteration and how we would classify it. If we believe that was actually the answer, we would report it out as a relevant finding on the front page of the report.
With your own classification?
With our own classification, yes. Mm-hmm. Independent to the previous classification.
Great. So it sounds like also if they've had a limited gene panel in the past, we'll re-review those genes as well, and we won't just rule those out.
Yes,
great.
That's right.
Okay.
It can be beneficial to do exome sequencing when you're not totally sure of the inheritance as well of a lot of these findings from panels, because we usually are doing trio testing and we can determine whether the VUS
is inherited from an unaffected parent. So it can be really informative too.
Great. One question came up here saying, would children of the proband, so this actually sounds like it's from a patient, my parents passed away and my most close distant family members passed away too. My doctor suggested sending my children's samples along with mine. Could that be considered an informative trio, and would that affect the sensitivity of the testing?
So generally first-degree relatives are considered an informative trio. We'd have to look at it more carefully at the particular family structure and the disease that's being inherited through the family. And we have clinicians oftentimes emailing us saying, hey, I have this family with this particular family history, and these are the individuals that are available for sequencing. Would you consider this an informative trio? And we're happy to answer those questions. But for the most part, first-degree relatives are considered informative.
Great. Let's see here. If this is another question about like dual diagnoses. So if a patient has multiple phenotypes, do you think this testing would be able to identify both those diseases?
Absolutely. I think that's one of the best benefits of Exome is that we can pick up these dual diagnoses, because traditionally clinicians will stop once they find a positive finding if they're doing single gene testing or panel testing. When you're doing exome sequencing, it's agnostic, so you're actually looking at everything. So I think that's probably one of the best benefits of exome sequencing is the ability to detect dual diagnoses.
Great. One question that came up is, what are your thoughts on DNA banking after an exome has been run? What is the best way to plan for future testing needs?
That's an interesting thought. I mean, I think we work in a DNA lab, so I think we all would say that DNA banking is a good idea. I think, the technology is improving. I think, really over time since the launch of exome sequencing, the number one factor that has improved is the number of gene-disease relationships that we understand today. That's why we have such a high rate of reclassifications. And so that we can do on the existing data. But there is always going to be improving technology, technologies that are improving and better than the last. And for patients that had a negative exome, we have this conundrum: what do they do next? And there are multiple research projects and genome sequencing and other methods. So I think I would say absolutely. If you have a negative result and you're just looking for to continue looking down different avenues, I think the technology is improving all of the time. We probably won't have any huge leaps like we did with Exome for a long time, but I think banking DNA is an absolutely good idea. Would you agree?
Yeah. Right. Some technologies are requiring fresh samples, so it may be harder to get RNA sequencing from banked DNA, but if that's what you have, definitely.
And I think also looking at the philosophy of the laboratory that you're doing whole exome sequencing with, often, maybe it's the technology that's changed, but sometimes it's just the data and the patient for life philosophy. I don't know if you want to talk a little bit about that, but the analysis that the laboratory does for future years is very important as well.
Yeah, definitely. So why don't you talk about the patient for life, since that's your forte.
Once you sequence the genes, the DNA, you can actually go back and reanalyze that information repeatedly as information about the genes changes. So as long as it's sequenced well and filtered well, you can go back and reanalyze data without actually using a fresh sample a lot of times. We might want the banked DNA to Sanger confirm an alteration, but the way that we actually use previous data, even from negative cases, is we always keep those alterations in our database and continually review them if a new finding could be relevant. So while it would be useful to bank the DNA, there's also information that's kind of banked in a way.
Great, thank you. One question came up, but I'm not sure I know the answer to this, or you guys know the answers to this, but the question was, how many de novo variants does the average human have?
It depends on if you're looking at the whole genome or if you're looking at the exome. And I believe if you're looking at the exome, I wish Shaw was here.
I know.
What is it, 1?
I think it's an average of 1.
Yeah.
But I mean, it also
exome.
depends on the age of the parents too. So it changes over time. Yeah.
Great. And then somebody asked, will the studies be emailed to us? So we are not planning to email those papers out, but they are all available. All of the ones that Ambry has worked on or collaborated on are available on our website. So if you go to our website under Research and Publications, you can find the links there for the papers. I think a lot of them are under clinical genomics, or they may be some of the other subsections, but they should all be there. And I think that is all of the questions. And if anything else came up, feel free to email us or contact us through our website, and we can get those questions routed directly to the panelists, and we'd be happy to answer them for you. Thank you, everybody, for joining today. And we will be posting this webinar on the website in the future, so you can always look for that as well. Thank you.
Thanks.
Thank you.